So I'm going to talk about lentiviral generation of CAR T cells targeting Kappa Myeloma Antigen, or KMA, for the treatment of multiple myeloma. And this is a collaboration between the Centre of Excellence and biotech company HaemaLogiX, which many of you will have been involved with the KappaMab antibody. So as we've heard, CAR T cell therapy and BCMA therapy is really now center stage in the relapsed refractory setting for multiple myeloma. But there's quite a lot of variation in terms of the response rate. They're high, but the duration of the response can vary depending on the different products that are being used. And it remains to be seen whether there's going to be a plateau on the curve or whether eventually all myeloma patients are going to relapse. So that's one of the really outstanding questions. Infection remains a major problem post-CAR T-cell and BCMA therapy, as we've already heard. And there are a number of new targets in development to try and combat some of these relapses post-BCMA therapy, and KMA is one of those that's being developed. Kappa Myeloma Antigen is a tumor-specific membrane-expressed antigen on malignant plasma cells, and it's restricted to Kappa type. There is a Lambda equivalent of this. And the antibody MDX1097, or KappaMab, is a chimeric antibody that binds to a conformational epitope in the membrane-bound antigen, which is less likely to bind to the circulating light chain, and also spares the lambda-expressing B cells, suggesting that you may have less immunopoiesis and less infection problems because you still have half of your B cells still around. KappaMab, as we know, is being investigated in a number of early phase clinical trials run by Andrew Spencer and HaemaLogiX. And we've been working with HaemaLogiX to generate a construct using the single chain variable fragment of KappaMab fused to a relatively vanilla CD28 41BB CD3 zeta chain construct. This started out life as a transposon construct at the Westmead group, and then we brought it into our group to transfer it into a lentiviral backbone. So what happens to the T cells when you transfect them? And essentially they proliferate really well, and there's some evidence to say that they proliferate even better than the untransduced cells in terms of cell numbers, but also population doublings. And we've got nice expression of over 70% in the CD4 compartment and over 50% in the CD8 compartment. And this is stably expressed over time. So you can see here we've got a tracking experiment where we start off with 65% of the T-cell compartment having a detectable CAR on the surface. And over time, this is at least stable, and there may be further enrichment with as the cells expand. The other nice thing to see is we have a really nice phenotype of these cells with a very high degree of stem cell memory phenotype, as opposed to effector memory, which is relatively exhausted. And this seems to improve over time with the cells in culture. And you can see here, this is also, you get stable expression of both the CD8 and the CD4, with probably an improvement in the CD8 fraction over time. And the stem cell memory phenotype, again, improves over time in culture, which is a little bit counterintuitive. You would have thought that over time, with the cells being in culture for longer, they may have gone into a more terminally differentiated state. But this is really exciting for us to see that we have a really nice product profile when we do eventually go into patients. So we've taken this into we've looked at the effect of these cells on KMA positive and KMA negative cells. There's no cross-killing for the negative OPM2 cells, whereas in the JGN3s that have been engineered to express a mid-phase level of Kappa Myeloma Antigen, they natively express it as well. You can see that there's a really a nice killing profile following a dilution effect with the target effector ratio. And if anything, the day 12 cultured cells seem to be the most effective cells, but they don't seem to fall off as they've been in culture for longer. And we have a really nice killing effect in the earlier cells that have been tested, which have been cultured to day 9. This just shows the interferon secretion of these cells. So we've got really nice interferon secretion at the 2 effector target ratios tested. And we've taken this into a mouse model. So the JGN3 KMA mid cells are placed in an NSG mouse. And then on day 7, after the tumor is established, they get CAR T cells at a couple of different doses, so 2.5 or 5 times 10 to the 6 cells infused, and then we do serial BLI imaging to track the tumor growth, and also we bleeding the cells to look at the mice to look at the CAR T cell expansion kinetics, and it's a survival experiment. And here we can see that on the left-hand side, the bioluminescent images with the untreated animals at the top, animals that have had untransduced control T cells in the next column down, KMA treated animals at 2.5, and then the bottom line of animals is the ones who've had 5 million cells. And you can see a really nice anti-tumor response. And there seems to be a dose response as well, with the 2.5 times 10 to the 6 cohort losing tumor control over time, although this, at the time this experiment is still ongoing, all of the animals are still alive. But there's tantalizing evidence in this experiment that the 5 times 10 to the 6 animals have been maybe cured. And we will be updating this data over the next couple of months, and we'll be repeating the experiment with parental KMA native expressing cells in the next cohort. So in conclusion, we get consistent and high expression of KMA on donor-derived T cells. The product shows a really nice phenotype. We're having effective killing in vitro, and we've got cytokine production on target recognition, and an evolving story to say that we get very good anti-myeloma control in the animal model system. This has been a really great collaboration between ourselves, the HaemaLogiX team, and also the team at Westmead who did the initial work on generating the construct, which has then been re-engineered by Jane and her team to take it into the lentiviral system. And most of the work I've presented today was done by Nicole Haynes and Jessica Lee. So I'm happy to take questions. Ajay. So the experiment's ongoing, so we will be looking at that in the future. Hang.
Show my ignorance.
No such thing as a stupid question, Hang.
Just wait. Is there a rationale of looking at its activity in a lambda producing myeloma cells? I'm trying to look at epitope spreading and whether or not otherwise you're going to just use this in a Kappa myeloma only.
So we will be doing those experiments as we progress down that line. There was some problem with that cell line, so we couldn't put that into this experiment. So yes, we will be looking at lambda expressing cell lines, partly to make sure there's no allo effect as well. Okay, if there are no more questions, we'll move on to the next abstract. So I'd like to introduce Mary Sartor, who continuing the KappaMab story. So Mary's a principal scientist in the flow cytometry lab at Westmead, and she's going to talk to us today about expression of KMA antigen, Lambda Myeloma Antigen on bone marrow plasma cells in patients with myeloma. Thanks, Mary.
Thank you. Firstly, I'd like to thank the organisers for giving us the opportunity to present our data. Today I will be talking about myeloma, Kappa Myeloma Antigen, which I will refer to as LMA, and also, sorry, I'll start again. Kappa myeloma, I will be talking about the expression of Kappa Myeloma Antigen, which I will refer as KMA, and Lambda Myeloma Antigen, which I will refer as LMA. As we heard from Professor Harrison in this previous talk, KMA is actually a potential target for immunotherapeutic approaches, and including CAR T-cell therapy. So the expression of LMA and KMA are actually found on the surface of plasma cells. They're also found on multiple myeloma cell lines and also on occasional nuclear cells in tonsils and secondary lymphoid tissues. What I need to stress is that KMA and LMA are not found on normal bone marrow or normal immune cells. In this study, I'm going to show you the results of two antibodies called KappaMab, which targets KMA, while the 10B3 targets LMA. Both antibodies are directed towards a conformational epitope within the free light chain, and neither antibody binds direct immunoglobulin. We used multiparameter flow cytometry to investigate the expression profiles of these antibodies, and the antibody panel consisted of the following markers. We used 269, which is usually referred to as BCMA. We used 38, 138 and 45 to actually gate for the plasma cells, and then we looked at the expression of KMA and LMA with 269 and 56 and SLAM F7. The antigen density for both KMA, LMA and BCMA was calculated using quantum-bright beads in PE. Now, this is a typical example of the flow cytometry gating that we used. This is an example of a bone marrow sample which was taken from a newly diagnosed lambda-restricted patient. So initially what we do, we try to investigate. Is there a pointer? No, sorry. I thought that was a pointer. Okay, we initially try to identify a singlets population by looking at forward scatter versus forward scatter height versus forward scatter area, which we see here in the first dot plot. We then look at CD45, which is on the y-axis, versus CD38 on the x. This allows us to capture all the nucleated cells by excluding debris and red cells, which we can see in that lower left-hand corner. We then identify our plasma cells by the co-expression of 138 and 38, which we can see in the very dark dots. The next dot plot on the left-hand side is actually looking at the co-expression of CD69 and 10B3, which is the LMA antibody, and we can see that the LMA is actually expressed quite brightly compared to the 269, which only has dim expression. The bottom right-hand dot plot is KMA versus LMA, and we've used KMA here as a specific negative control for LMA. And as you can see, there is no staining of the KMA. So this slide here represents a summary of our data on the newly diagnosed and relapsed multiple myeloma patients. As you can see, in the newly diagnosed, we looked at roughly 23 patients, and CD269 or BCMA was expressed in roughly 90%, whereas 2/3 of the cases expressed KMA. In the relapsed refractory group, we find that the 269 is actually reduced in proportion, in percentage, whilst KMA is actually enriched compared to the newly diagnosed portion. Within the relapse refractory, we were also able to identify 2 patients that were KMA positive but BCMA negative. We also looked at SLAM-F7, which is CD319, and found that that particular antigen was expressed in all cases. When we looked at the antigen density, we found that in the newly diagnosed cases, both KMA and BCMA had relatively similar antigen density, but within the relapse refractory, we found that BCMA was remained fairly stable, whilst KMA was increased. So this is an example of a KMA positive plasma cells in a patient that has been treated, that has multiple myeloma, that is now on maintenance therapy on lenalidomide after stem cell transplant. As you can see in these slides, there are very few plasma cells, roughly less than 0.1%. But nevertheless, we can see that there are 2 populations. If we look at the second plot on the right-hand side, it's showing 2 populations of plasma cells, whereby at least 60% of these are KMA positive. If we look down at the bottom left-hand dot plot, we find that these plasma cells are actually 269 or BCMA negative. On the right-hand side, we have KMA versus 56, and 56 is expressed in all cases. So looking at the summary of LMA, once again in newly diagnosed and relapsed refractory multiple myeloma, we find that the expression of LMA both in the newly diagnosed and relapsed refractory is roughly about 50%. I must admit here the numbers are quite low, but the pattern's there. And whereas in 269 or BCMA is expressed in the majority of cases. We then looked at the antigen density within these 2 groups, and found once again that LMA and BCMA in newly diagnosed had equivalent antigen density, while in the relapse refractory, the BCMA was reduced, but the LMA was increased within the actual samples that we were able to, within the samples that actually expressed the LMA. So this is an example of the antigen density of LMA, once again, relapse refractory patient. And if we look at the first dot plot, we can see BCMA versus LMA as described by the 10B3 antibody, and we find quite bright expression of the LMA staining, whereas BCMA has relatively dim staining in comparison. If we look at the bottom right dot plot, we find that 56 is negative on the plasma cell population. So we also looked at KMA expression on early multiple myeloma. On early plasma cell dyscrasia cases. For example, this one here is an example of MGUS, and this the first dot plot on the left-hand side is actually showing 2 populations of plasma cells. We have the light grey, which are 45 positive and 38 bright, which we consider to be a fairly normal population of plasma cells. And we have a second population, which is 45 low and 38 low, which are the malignant plasma cells. If we look at the next dot plot, plot B, we find that KMA is actually expressed on the abnormal plasma cells, whereas there's lack of expression on the normal plasma cells. And if we look at 269, 269 is actually not very dim. If expressed, it's very dim. So here we find that we have quite specific expression of the KMA on malignant plasma cells, but not on normal cells, and BCMA is negative on both. So in conclusion, we can say that KappaMab and LambdaMab, being 10B3, are specific antibodies that target KMA and LMA found on malignant plasma cells, multiple myeloma cell lines, and occasional secondary lymphoid tissue. But they're not found on normal plasma cells or other normal immune cells. The antigen density of KMA and LMA is enriched in the relapsed refractory group relative to the newly diagnosed samples. While BCMA density is decreased or unchanged, it may indicate a treatment-resistant clone. So, and finally, KMA and LMA represent valuable therapeutic targets in relapsed refractory multiple myeloma, with a combination of high antigen density and target restrictions to the malignant plasma cells. By targeting LMA and KMA, it could provide an advantage in the sense that the normal plasma cells remained intact, and this could actually reduce the toxic effects that we see, particularly with hypogammaglobulinemia and infections. Thank you.
Thanks, Mary. Great presentation. Questions for Mary? Miles?
Thanks very much. The question I've got relates to just understanding what this antigen does and where it develops in the lymphocyte plasma cell transition. So, what is the expression of this antigen in uneducated lymphocytes, lymphocytes that have not been through the lymph node during isotype switching, and the point of it is where with this, the antibodies, how far back in the lymphoid maturation process do they hit?
Yeah, okay. The antibodies aren't expressed on normal B cells. Okay.
Oh, sorry, yeah. So, but
where in
that, at some point they become a plasma cell doesn't suddenly go from a...
Yeah, I know. It starts from activated B cells, yeah. I think Roseanne might like to comment on this. Sure, come up here. Yeah.
So, yes, that's always a very good question. So the fact is that the antigen it originates from an activated B cell. And this goes back to the work that Bob Brazen did in Tony Baston's lab, and Chris Goodnow as well identified KMA initially. So what happens is if you activate a naive B cell with, say, LPS or something else that activates a naive B cell, you get KMA expression on the surface of the cell. And this goes back to the original K121 antibody. So you get KMA expression on the surface of the cell in a naive B cell. Also, we subsequently looked at if you take the typical cytokines for T-cell activation, for example, an anti-mu, IL-21, etc., the standard activation cytokines for naive B cells, you can get both KMA or LMA expression on the surface of the naive B cell. So the activation of the B cell then obviously indicates that it's going to go through maturation. So it's going to go through the germinal center, maturation, it's going to be selected and then obviously hypermutated, and you're going to get, select your antibody. Why KMA and LMA survive that process, whereas a normal B cell doesn't, is not really known. But in fact, what we have found is that if you look at immunohistochemistry, of all normal normal human tissue, so all tissues, including, say, the gut, and of course the lungs, etc. There are occasional KMA or LMA expressing B cells in the secondary lymphoid tissue, in the mucosal tissue, but mainly in the tonsillar tissue as well. And this has also been shown. So what are these antigens? I mean, obviously they're something different because the antibodies select them because it's a conformational epitope that is found in the constant region. It's easy for, for Kappa because it's got one constant region. Finding an antibody that binds to all three isotypes of the lambda, the 1, 2, and 3, was actually difficult, and we did that obviously with transgenic mice, so that's how we discovered those antibodies or created those antibodies. So why are they in the membrane? What is holding them there? It's actually sphingomyelin. So if you prevent the breakdown of sphingomyelin, you actually increase the expression of KMA. And what is sphingomyelin? So it's that pathway where you break down sphingomyelin in apoptosis, and you create ceramide and sphingosine, and ceramide is the apoptotic pathway. So what these antigens are doing is they're being held in the membrane with sphingomyelin and presenting this epitope. For some reason, apoptosis does not occur in the myeloma malignant cells. We found them in amyloidosis cells as well, in particular LMA. So they're very unique to a malignant plasma cell, but also to an abnormal plasma cell. We have actually found these in SLE and RA as well. Yes.
Thank you.
Sorry, did you want?
So I have a question.
So last question.
Okay. So have you looked at the KMA and LMA expression after treatment with like bortezomib or lenalidomide?
Yeah, that's exactly what Mary did here with the
Okay.
relapsed refractory.
Okay.
So in fact, Mary in David's lab has actually found that post-treatment of an IMiD, not specifically, but of, say, a proteasome inhibitor or an IMiD or a stem cell transplant, the antigens KMA and LMA are actually, the density of them is higher than BCMA. So that was what Mary showed, yeah.
Unfortunately, sorry, I was going to say, unfortunately, within the LMA, we didn't, I mean, the patient cohort was quite small, but even though there were only 4 patients within the relapsed refractory group, 2 of them were actually positive. And if you looked at the antigen density of those 2 particular patients, it far exceeded the antigen density of a BCMA. Yeah. So it's quite
Yeah, and I think the Westmead group are looking for people to provide samples of patients to try and expand the data set, and the number of us are going to be helping with that. So if you've got further questions or you would have a sample set, I'm sure David and Mary would be delighted to talk to you in one of the breaks.